So you want to make sourdough bread. Maybe you've heard of the health benefits of natural fermentation. Maybe you like the look of artisan bread. Maybe you just like playing with dough. No matter the reason, we're here to show you how to make sourdough bread and show you some of the cool microbiology behind sourdough. Making a sourdough starter is surprisingly easy. Simply mix equal parts by weight, flour and water in a covered jar. 100 grams to 100 grams is a good place to start. Leave the resulting mixture out of direct sunlight at a consistent room temperature. Feed the mixture every 24 hours, again with equal parts flour and water, and after 5 to 7 days, native lactobacilli and yeast will most likely become dominant. At this point, the starter will smell slightly fruity and faintly of ethanol with a foamy surface. After this stage, the sourdough starter is ready.
During the creation of the sourdough starter, we analyzed the solution and environment of our sourdough starter by plating it on several different media types. We did this with the purpose in mind of confirming that what we thought was in our sourdough starter was actually in our sourdough starter. On top of plating our sample, we observed some of the colonies that grew in our MRS plate, which would allow us to see the cellular morphology of the lactobacilli that we would grow. What we found confirmed our expected results from the MRS plate. Using a differential staining technique called a gram stain, we were able to identify gram-positive rod-shaped bacillus cells. This coincides perfectly with the profile of Lactobacillus sanfrancensis. When you combine the results of our plating as well as our observations from the gram stain, we're pretty comfortable in saying that lactobacilli are responsible for the unique taste we found in our sourdough bread via heterolactic fermentation. So in order to understand why the processes that our microbe goes through are important to the bread making process, we have to first describe fermentation. There are two types of fermentation that are relevant to L. sanfrancensis, and the first would be homolactic fermentation. Homolactic fermentation only produces a singular product, that would be lactic acid. Then the second type of fermentation would be heterolactic fermentation. Note that these are exclusive. If one is homolactic, it can only produce lactic acid, whereas if one is heterolactic, it produces lactic acid, but at the same time it could produce another product such as carbon dioxide. Please note that homolactic fermentation bacteria are typically more used in pickling, as they typically only decrease the pH of the solution they're in, so that would be useful in brine solutions. But in baking solutions, heterolactic fermentation is more useful in sourdough, as it produces more products and leads to different tastes and different aromas.
Why do lactobacilli and yeast consistently come together to dominate sourdough starters across the world? Lactobacilli and yeast gain a foothold in sourdough starters because they are commonly found in nature. They become dominant since their strong symbiotic bond allows them to outcompete most other microorganisms in the starter. The conditions for their symbiosis are established as lactobacilli acid metabolism byproducts begin to acidify the starter. These acid conditions inhibit most microorganisms, but not acid-tolerant yeast. With most of the competition out of the way, yeast and lactobacilli come into their own. In some starters, yeast do not metabolize maltose, a sugar favored by some lactobacilli, which further supports close connections. The fact that the same relationships emerge across the world indicates the strength of the lactobacilli-yeast bond. When combining the ingredients for the dough, first add the starter to the water and mix until combined. This distributes the lactobacilli and yeast and leads to a more consistent rise. After the starter and water are mixed, add the flour and mix until the dough is combined into a shaggy ball. Now that the sourdough starter has been made, it is time to make sourdough bread. About 4 hours after feeding the starter, it should be bubbly and float on water, indicating that it's ready. Once the starter is ready, we can begin making the dough. Various water to starter to flour ratios can be used, but we use a simple 2-part water, 1 part starter, and 3-part flour, with a little bit of salt for flavor. After the initial shaggy ball has sat for about 10 minutes, begin kneading the dough. Stretching and folding the dough over itself is one of the better methods of kneading. Do this for about 5 minutes, let the dough rest for another 10 or so, then stretch and fold again. For best results, do this 4 to 5 times, but one thorough stretch and fold session works almost as well. After the kneading, cover the dough and allow it to bulk rise at room temperature for about 3 hours. After the bulk rise, flour a work surface and form the dough into a ball by stretching the top surface and tucking the excess underneath. After the ball has been formed, transfer the ball into a bowl. Either flour the bowl or set a flour towel into the bowl to make removal easier. Cover the dough and allow the dough ball to rise overnight in the fridge. After all the hard work making the dough, it is time to bake. Preheat an oven with a Dutch oven inside to 480 degrees. If you don't have a Dutch oven, you can use a lidded pot to contain the steam in the oven. After the oven has preheated, transfer the dough ball from the bowl into the heated pot or Dutch oven. Cut the surface of the dough with a sharp knife to allow a place for the dough to expand without randomly breaking the skin of the dough ball. Get creative and carve a jack-o'-lantern or other shape like we did. Replace the lid and return the pot to the oven, and allow the bread to bake covered for about 25 minutes. After that time, remove the lid, reduce the temperature to 420 degrees, and allow the top of the bread to brown slightly. This may take another 20 minutes or so.